Hello everyone and welcome to the Picture Education. It's the lecture 3 on the biochemistry of acid and base. So coming to the acid and base, acid is a moiety which can release a proton in water and the base is a moiety which can accept proton in water. So why I am talking in water is because cell has a major fraction as a water, and the molecules and the biochemicals that are present are in the aqueous environment of that cell. So if a molecule can release a proton in that environment is known as acid, and a molecule which can accept a proton that is released by acid is known as base. So the water can act as both acid as well as base. So this property gives water a amphoteric nature. We are going to talk about the amphoteric nature in upcoming slides. But there is also a term called a strength of an acid, which is defined as the tendency of a molecule to release a proton. So if a molecule can easily release a proton, it is highly acidic in nature. So coming to the reaction of a water, so water can self-dissociate into the proton as well as hydroxide ion. So this reverse self-dissociation is also known as ionization, which generate a proton as well as hydroxide ion. But the hydrogen atom do not exist as free uh, proton, but actually they make a complex with the water and forms a hydronium ion. So we are going to use the law of mass action, and we will define the equilibrium point of the dissociation reaction, that how much water molecule can dissociate into the concentration of the proton or to the concentration of the hydroxide ion. So coming to the equilibrium constant, so K equilibrium is defined as the concentration of the product by the concentration of the reactants. So here the concentration of products are the concentration of the proton into the concentration of the hydroxide ion by the concentration of the reactant. Here H2O is a reactant while the H plus and OH minus ions are the reactant. So K equilibrium equals the concentration of proton into the concentration of hydroxide ion by the concentration of water. So these square brackets are representing the concentration which is in moles per liter, which are designated as the capital M. And the K equilibrium was determined experimentally and it found to be 1.8 into 10⁻⁻16 molars at 25 degrees Celsius. And we can easily determine the concentration of pure water just by taking a 1 liter of water, divided it by a molecular weight of water, which is 18, and we get a concentration of pure water to be 55.5 molars. So we just rearrange the equation, we just multiply the water with the K equilibrium, we get H plus concentration into the OH minus concentration equal to K equilibrium into the concentration of water, or the proton concentration into hydroxide ion concentration equal to K equilibrium into the water concentration. So we just substitute the value from the previous slide, we get the concentration of a proton into the concentration of hydroxide ion to be 1 into 10⁻⁻14 molar square. So from this equation we found that the one molecule of H2O is uh, ionizing into the one atom of the proton and one molecule of the hydroxide ion. So the concentration of H plus and OH minus must be equal. So we just substitute the OH minus with the our proton concentration. So which we found to be proton concentration to be 1 into 10⁻⁻7 molar. So pH is defined. So this particular equation actually help us determining the pH of the water. So pH is defined as the negative log of the H plus ion concentration. So just by substituting the value, we get the pH of water to be equal 7. And by the previous equation, we know the concentration of proton into the concentration of hydroxide ion, which is equal to 1 into 10⁻⁻14 molar square. So if we take the negative logarithm on both sides, we get the pH plus pOH to be 14. So pH scale actually ranges from 0 to 14, and in that scale the 7 is neutral, where 0 is highly acidic in nature while the 14 is highly basic solution, while the 7 is neither acidic nor basic. Coming to the conjugate acid and base concept, if we take a acid which can dissociate into the proton as well as conjugate base, so what is a conjugate base? This is the question. So the conjugate base is the base formed by the removal of a proton from an acid. So when an acid actually removes the proton, is known as conjugate base, and the conjugate acid is the that removed proton is known as conjugate acid. So the acid and the conjugate bases are complementary species. So every acid will have a conjugate base. So for example, if we take acetic acid and react it with the sodium hydroxide, we get a sodium acetate, which is our conjugate base, plus water molecule, which is our conjugate acid. So just like water, we can define the dissociation constant for this acid. So Ka must be equal to the concentration of the product by the concentration of reactant. The product are the H plus and the conjugate base, and by the concentration of the reactant. So K equal to concentration of proton into the concentration of conjugate base by the concentration of the acid. So just like pH, we can also define a pKa. So pKa will be the minus log into the concentration of a Ka. So if we have a smaller pKa value, it will mean the compound or the molecule is a strong acid. If we get a larger pKa, we will get a compound or molecule to be a strong base. So we are going to use this concept in upcoming buffer lecture, and will define that how we can use the dissociation constant and help determine the buffering capacity. So thank you for your patient listening, and stay tuned for the upcoming lectures. Thank you.